Okay, so here you are back in our experimental design and analysis lecture 6, which is about the assumptions of parametric statistical tests, and part 2 of that lecture is about the assumption of normality, that there is an underlying normal distribution in the case of a t-test and many other parametric statistical tests. So we're going to look in particular in this section at how to deal with situations in which the data are not normal, and one way that we can deal with it is to use the transformation, a mathematical transformation, to change the shape of the distribution until it is normal and then perform a t-test as planned. So just a reminder, this is the third crucial assumption that we're making in our parametric test. First was that the observations were independent. That's what we dealt with in the previous lecture. The second is that the variance is homogeneous. I dealt with that in the previous part of this lecture. And the third key assumption is that the data come from a normal distribution, a continuous, because normal distribution is for a continuous variable, a continuous normal population. So in other words, there should be some kind of bell curve for the measurement that we're looking at in the population that we're studying. So let's imagine that we've collected some continuous data, so there's a chance that they're normally distributed, but when we look at the distribution we find that they're not. And here's an example. We might measure the area of the leaves of some seedlings on the forest floor, and I've plotted here leaf area against frequency, so this is a frequency histogram of leaf area, and although it's sort of a bit bell-shaped, bell curvy, we've got a definite lopsidedness to this distribution. There's a long tail on the right and the data are kind of squidged up against the left-hand side. And that's because leaf area can't fall below 0, and yet the mean leaf area is quite small because these are little seedlings on the forest floor. But there are a few seedlings with very large leaf areas, which have obviously grown more than the other seedlings in the study. So you look at this distribution and just eyeballing it, you think it doesn't look hugely normal. It's not terrible, but it doesn't look that normal. At that point, you might just conclude, right, well, let's get on with dealing with it. But of course, we'd like to try to be objective where possible. So we're going to run a statistical test, you guessed it, to find out whether the data are indeed significantly different from normal or whether actually they're roughly normal in the, after all. So visual inspection gives us an idea, but there's a test called the Shapiro-Wilk test, which will actually give us a more definitive answer. The Shapiro-Wilk test, it's another of these statistical tests named after some crusty old statisticians. It's just a shame, really, that we have to learn all these awkward names, but it's a test for normality. So the null hypothesis in a Shapiro-Wilk test is that the distribution of the data is not different from the normal distribution. And obviously, if we have to reject that null hypothesis, then we've violated the assumption of the t-test and we've got to think again. So the Shapiro-Wilk test, just like all other stats tests, generates a test statistic. In this case, it's called W. Previously, we saw F and we saw T. And once we have the test statistic, the mathematical boffins have figured out how that test statistic behaves under the under the null hypothesis. And so when we look at our value of W that we measure from our data set, if it's out of line with what we'd expect under the null hypothesis, then we're going to see that it's very unlikely under the null hypothesis. In other words, we're going to see a low p-value. So the computer will do the work for you. It generates a test statistic, W, and it generates a p-value, in this case another small p-value showing a significant departure from normality. So we've rejected the null hypothesis of the Shapiro-Wilk test. The distribution of the data is not normal. So what do we do? We've got a distribution which looks wonky, and indeed the Shapiro-Wilk test confirms that it is wonky. But the t-test assumes it's not wonky. It assumes the data are nicely bell-shaped in their distribution. So we've got a couple of options here. One is we could try to force the data to behave in the way that we want them to behave. So we could take this distribution of leaf areas and try to do something to it which makes it look more normal. And one way to do that is just to apply a simple mathematical equation to each data point. And these are often very simple transformations, but they can produce nice changes in the distribution which generate much more normal looking data. And in this case, I deliberately chose leaf area because it's a very obvious transformation that we can apply. As plants get bigger, their leaves get longer and the area of those leaves gets bigger as a result. But the rate at which the area gets bigger is much greater than the rate at which the leaves get longer. There's a change that basically the area will increase with the square of the length of the leaf, for obvious reasons, because area is measured in square units squared, and a leaf is not a sort of two-dimensional object, it's a three-dimensional object. It's got width and length as well as height. And so an obvious transformation here is actually to take the square root of the area so that we end up with something which is measured in units that are sort of equivalent to the length of the leaf, and that may follow a normal distribution. It turns out quite often in these sorts of situations with areas in biology, that's what happens. So if we square root the data, turn it into a frequency histogram of square rooted leaf areas, and we get suddenly a much more symmetrical looking distribution. It's actually very bell shaped. It looks like a good fit to the normal distribution. And indeed, if you then run a Shapiro-Wilk test on that second set of data, once all the data have been square rooted, you find that there is no longer a significant departure from normality. So we would conclude that the square root data are normal, and we can go ahead with the t-test or whatever it is we want to do with these data. So this is a nice illustration of when a transformation works, but of course it's not always obvious what transformation might work, and it's not always possible to find a transformation that does work. But just to give you some other examples, I talked about the square root transformation. You might often want to do a cube root transformation if you're measuring volume, because volume will tend to scale with the cube of the length of something, and the lengths might have a normal distribution even if the volumes don't. But other transformations you'll see in the literature, or when you're doing analysis for practicals and projects, you might come across the log transformation, which is commonly used in certain circumstances, and even something called the arc-sine transformation. So I should say the log transformation has a fairly similar effect to a square root transformation. It makes the distances between big numbers relatively small when you perform this transformation. So basically it squishes up the right-hand side of the distribution and it stretches out the left-hand side of the distribution. Whereas this transformation, the arc-sine transformation, so you actually do the arc-sine of the square root of the data, tends to be a good transformation to use when you are looking at percentage or proportion data. So if you don't recognise arc-sine, I don't blame you. It's a fairly obscure trigonometric function. You might be more familiar with sine, cos and tan. Well, arc-sine is another trigonometric function which does similar things. And in the case of percentage data, it can help normalise them. So if we look at this data set here, this is a frequency distribution for the percentage expression in cells, samples of cells. So let's say we've got a whole load of Petri dishes, we're looking down the Petri dish at a whole load of cells that are being cultured, and some of those cells are glowing green because they're expressing a protein of interest. Some of them are not glowing at all or only slightly, and that might have some significance in the biological system we're studying. So we can measure for each Petri dish what the percentage expression is and then plot that as a distribution, and we might well find a distribution like this, which is left skewed. It's got a long tail on the left and it's squidged up against the right here, and that's because at the right hand edge here we've actually got 100%. Once you get to 100%, you can't have more than 100%. If all the cells are expressing the gene or whatever it is you're interested in, then that's the maximum. So this kind of left skewed distribution is reasonably common with percentage data, and an arc-sine transformation might sort it out. Here we go, I've arc-sine transformed the data, and hey presto, it looks rather more normal than it did before. Okay, so I wouldn't sweat too much about remembering these particular transformations. What I really would like you to take home from this is that a key assumption of many parametric tests is that the data follow a particular distribution, and more often than not we're talking about the normal distribution. And quite often data do not fit with that assumption, but there are things we could do to try to fix that problem, including transformation. But sometimes transformation doesn't work, and that's when we come to needing to use an alternative test which doesn't make the assumption of normality. And so that's what we're going to deal with in the next part of this lecture. We're going to look at non-parametric difference tests, a non-parametric equivalent of the t-test.